So it seems like physicians, as well as women, are interested in a drug that may have slightly less efficacy, although it's not clear. In the phase 3, we're trying to prove that we have similar efficacy, but no really untoward effects that are, at least at the moment, being described in relation to the drug. Current development from a regulatory point of view, we got the approval from the FDA in relation to the CMC. This is really critical since Minerva is manufactured from botanical sources, and it's the first drug that the FDA went on record to approve the CMC for a phase 3 oral drug. They approved already one topical drug all the way to the NDA from a botanical source, but this is the first oral drug that the FDA approved the CMC for. So that shows that the FDA is really interested in such drug, as well as that Bionovo was able to really control the manufacturing to the point where the FDA was satisfied. The second thing, which was really critical for this type of indication, was that the FDA and the EMA accepted our clinical development plan. One of the questions that everybody wanted to know is, in light of all the risks that estrogen has in this particular area, and the fact that the SNRI seems to show other risks associated with this kind of treatment to a slightly older female population, what will the FDA require from the point of view of safety analysis prior to approval? And as I'll show you in a minute, actually the FDA said that our drug so far shows sufficient safety that the number of patients that are required exposure are 1,200 patients for 3 months for the efficacy study, and only 680 patients for the phase 3B, which is a 1-year study. So it's very small numbers for such a large indication for women that are going to be using this drug for a long exposure. Of course, if we find any risks during those 3 months or 1-year exposures that were not really described before, probably we will have to go back to them and see how we address them prior to an NDA. But as far as it looks right now, from the safety profile that we have and the agreement that we have from the FDA, it is not going to be a long, drawn, expensive development plan for approving Minerva. So I think this is really critical for investors to know that practically we have a strict timeline and a fixed cost to get this drug to an NDA, and it's not in the hundreds of millions of dollars, it's really in the tens of millions of dollars to get a multi-billion potential drug. The one question that everybody had in relation to that is, well, we know that estrogens work for hot flashes, but how do we know that a selective drug, an alpha or beta selective drug, will work on the menopausal hot flashes? And there's really no animal model to do that other than those 2 primates in women. It's easier and cheaper to do clinical trials than to get those primates and create the models in the primates. So one of the things that we did is to look at primary neurons in order to see whether it's the alpha or the beta effect. So we took embryonic stem cells and differentiated them along the lineages of cells, the neuronal cells that are related to thermal regulation, and we looked at both the genomic and the non-genomic effects. The genomic is really an effect that modulates the tissues to have a response different than the current response, and this data is really directly on the non-genomic effects. I can come up with the genomic effect as well, but this is more critical for a quick effect on thermal regulation, and that is measured by calcium oscillation. So what you can see is that, number one, those cells, when they're differentiated, express native ER alpha and ER beta, meaning that we do not have to artificially put the ER alpha and the ER beta into those cells. So in the brain, those cells will have ER alpha and ER beta. And when you treat them with estradiol, E2, you get a dramatic or statistically significant effect at increasing both the oscillations of calcium as well as the number of cells that are oscillating. Same thing occurs with MF101 or Minerva. But it doesn't happen with an ER alpha selective drug called PPT. So we looked at also if we increase the number of ER alpha copies, does that change the effect? And it didn't, which suggests that really the thermal regulatory effect is really mediated through the ER beta. All you have to have is a drug that really enters this particular part of the brain, that is thermoregulatory zone in the hypothalamus, which is not a given with every ER beta compound. You can have other ER beta compounds that are addressing the neurons or are interacting with the receptor, but not all of them, and not necessarily their immediate structural change will result in this type of effect. So there may have been some other failures. We know that Merck had a failure with their ER beta compound. The reason is it's not as good as Minerva. Minerva reaches the tissue and has this direct effect. And then, of course, we can talk about the safety of the alpha and the beta, but what you can see is that definitely Minerva does not have an effect on the uterus or on breast from the point of view of breast cancer development, and that is not just artificial. If you look at the human cells on the left-hand side, we do not turn on the oncogenes that are related to estrogen type of manipulation or type of increase of cancer risk. So why do we think that the drug is really worth developing? This is really an outcome of the phase 2 study. We conducted a phase 2 study that is very similar to the requirements of the FDA for phase 3 studies. So the randomized placebo-controlled study conducted in postmenopausal women that had 7 or more hot flashes a day or 50 or more a week, which is the FDA requirement. 217 patients in 6 centers in the U.S., and the principal investigator was Deborah Grady, who was really the first to show the cardiovascular risk of estrogen. 2 doses against placebo, and the primary endpoints that are mandated by the FDA are looking at the subjective diary of the frequency of hot flashes and the severity. Severity is a score that women get a definition of what is a mild, what is a moderate, and what is a severe hot flash, and then we give it a numerical value, 1 for mild, 3 to severe, and you multiply the number of hot flashes with the severity of the hot flashes, and you get a subjective score. So this is really the important aspect of the result. We had a statistically significant result, reduction in the hot flashes with a p-value of 0.04, 62% reduction in the moderate to severe hot flashes, and the odds ratio of 60% reduction on Minerva versus placebo was 2.4 with a p-value of 0.02, and I'll explain why that is significant for the phase 3. And also you can see that there is a clear dose response at the 12 weeks, and that's why we're going with a slightly higher dose in the phase 3 in order to achieve greater efficacy, because we were able to calculate what that dose will be in order to have similar effects to estrogen, somewhere around the 70 or 80%. From a safety point of view, which is very critical for this particular indication, we saw no difference in the number of uterine bleeding between treatment and placebo. It was around 9%. This is what you see in other estrogen trials. With estrogen, after 3 months of treatment, you see 50% abnormal uterine bleeding, so that's a very significant effect. We saw no cases of endometrial hyperplasia. With estrogen studies, you see 15% endometrial hyperplasia, which is the precursor to cancer. And I think that one of the most important things is there's only one single side effect, which we're trying to mitigate with the new formulation for the phase 3, where we removed some of the complex carbohydrates from the drug in order to remove the soluble fiber that may have increased the loose bowel episodes. Other important things, it seems like this drug is really affecting the homeostasis of an aging woman, and you see a statistically significant reduction in both weight and body mass index, as well as a trend towards lower blood pressure. It's important to associate those two because most drugs that are affecting body mass index and weight tend to actually increase blood pressure and cardiovascular risk, and in this case, that's exactly the opposite. And we understand the metabolic effects. I will not go into details right now, but it definitely seems like the estrogen receptor data is associated with a slightly different metabolic trend that results in this effect. The drug was very well tolerated. Again, very important for a quality of life drug. Other significant effect is, unlike gabapentin and Prespeak, Minerva, like estrogen, improves sleep, and there was a dramatic reduction in nighttime awakening. Those are the most significant hot flashes, disturb women so much so that they wake up at night, and we had a 67% reduction with Minerva. So the design agreed upon that we are initiating soon is a 1,200 patient study, randomized placebo-controlled, 400 patients per arm. There will be 2 doses, the similar dose to the higher dose of the phase 2 that has statistically significant effects, and a 2 to 3 times dose, which we will test the tolerability of it first in the 400 before we initiate the 400 patient trial for this particular dose against placebo. Very important when you look at the transition from phase 2 to phase 3 is to see what were the differences in trial design, so you can actually speculate on what kind of outcome you will get. Many times, phase 2s are very different from phase 3 because you're looking at in the phase 2 to just see a signal. This time we're actually looking at exactly the same type of effect. So the differences are we reduced the mass of the drug by 50% by removing those complex carbohydrates. We did studies to show that the pharmacokinetics and pharmacodynamics as well as the ADME is not different from the new to the old. The safety, when you increase the dose, you have to be concerned about what will be the safety. The no adverse effect level is 3 times higher in the dogs, 5 times higher in the rats. And just to give you an idea, with estrogen, it's 100% lower than human dose. So we are actually way in the safety range compared to other drugs in this area.